All right, everybody. So today's lecture is going to be on neurodegenerative disorders. This is one of my favorite lectures to give. So here at UCF, we have two classes called clinical neuroanatomy and clinical neuroscience. And in both of these classes, which are higher level neuroscience classes, we talk extensively about neurodegenerative diseases, their genetic components, their treatments, and then the future research and the future possible treatments that we're looking at right now. So let's look at a couple of neurodegenerative disorders. We don't have enough time to talk about all of them because there's way too many to count. But we are going to talk about the main ones. So this is Huntington's disease, Alzheimer's disease, ALS or amyotrophic lateral sclerosis, Parkinson's disease, and we're also going to dive a little bit into prion diseases, which are super, super interesting. So let's start off with Huntington's. So Huntington's disease, or HD, it used to be called Huntington's chorea. And chorea is defined as the spastic, uncontrollable movement, which is characteristic of someone with Huntington's disease, especially early on. Now, the name was changed to Huntington's disease instead of Huntington's chorea because in juvenile Huntington's, chorea is actually bypassed. There's no chorea whatsoever. So now it's just called Huntington's because we found that it's the same root cause. Now, this is an inherited disease. It's an autosomal dominant trait. And the first signs of this disease actually happen at the age of 35, which is crazy to think about because let's say that there was a person and they had no idea that they were genetically predisposed to this disease because of a parent that they had. Now that means that a person can live their entire life, including going to college, maybe even graduate school, medical school even, get married, have kids, and then get struck by this disease at age 35. And remember that this is a progressive neurodegenerative disease, which means as time goes on, this disease will get worse. Now, this affects 1 in every 10,000 people or so. And the more we talk about it, hopefully the less people this will actually affect. Now, in families with a history of Huntington's, this can actually occur earlier. So, and we'll get to why that is in a second. But the more this disease kind of goes on, potentially mutations will lead to this disease starting earlier in life. Now, in the United States, about 30,000 people have full Huntington's disease, whereas 35,000 people have significant symptoms. 75,000 people actually carry the gene, which is crazy to think about. Now, Huntington's disease, the gene that is in relation to this disease specifically, is on chromosome 4. It's on the P arm at location 16.3. So just something to think about. I would recommend memorizing where this gene is. Now, the meat and potatoes of this disease. So, the signs and symptoms. Initially, you're going to see a couple of coordination impairment. You might see a little bit of dementia. So, remember that Alzheimer's is the most common form of dementia. So, Huntington's disease incorporates a little bit of the same kind of symptoms, which is psychosis, or not being aware of your surroundings, or not knowing where you are, and not acting normally. Cognitive disorganization, and even personality change. So those are just some of the initial symptoms. Cognitive disorganization is in reference to cognition, which is the ability to think. So a person who has this is going to show with unstructured thoughts, the inability to focus, and things like that. Okay. Now, eventually, the muscle movement is going to be impaired. That includes both voluntary and involuntary movements. So that's going to be what chorea was, what I was talking about before. So these are jerking movements. It can also be slurred speech because, again, your muscles of the face, the mimetic muscles that are innervated by cranial nerve 7, are also going to be affected. So you're not going to be able to speak as well or as fluently and as fluid anymore. And there's also psychiatric problems such as depression, delusions, so a little bit of schizophrenia-like, and uncontrolled rage. And it's not too difficult to imagine that a person who has uncontrolled movements now and can't speak as well as they used to would get depressed. It's also not difficult to see how a person like with these symptoms can, lash out in anger. So imagine not being able to express your thoughts or having, hiccups in your thoughts. That could lead to a lot of buildup of rage and frustration. Now, later in life, patients will actually become mute. They'll lose their ability to use their vocal cords. They'll use the ability to use their mimetic muscles and their facial muscles, and so they just won't talk anymore. Now, cognitive non-function means that basically their ability to think and solve problems and interpret things is diminishing completely, and they will be immobilized. So people with Huntington's will eventually end up in wheelchairs. They'll get rigid joints because of these severe contractions, and they just won't be able to move. Now, keep in mind that someone with Huntington's, because of chorea, because of these jerking movements, they'll be constantly fatigued. And these movements don't even stop when they sleep. So even when a person with Huntington's is asleep, they'll still be spastically moving. Now, a lot of neurodegenerative diseases, actually, the way that a lot of people will die from them is actually due to pneumonia. And we're not really going to talk about the in-depth reason why. But another reason why someone with Huntington's may die is because of a choking hazard. So there's smooth muscle that lines your GI tract, starting from your esophagus. So in your esophagus, there's smooth muscle, and the esophagus is going to push down the bolus. Bolus is just the food that you've just chewed up, mixed with your saliva and those enzymes in your saliva. The esophagus is going to push that food down via wave-like contractions called peristalsis. So if a person is slowly losing their ability to control their esophagus and their digestive tract, this is part of the autonomic nervous system, they will likely end up choking. Also consider that a person who is unable to move their facial muscles and also their mastication muscles are not going to be as efficient at chewing their food. And so if you swallow a whole piece of bread or a whole piece of meat, you are more likely to get that lodged in your throat, in your pharynx, in your hypopharynx, and that can occlude your airway. So that can lead to choking and other hazards. Another reason why is heart failure, and unfortunately, another reason is suicide. Now, pathological features of Huntington's. So there's going to be selective neuronal degeneration throughout the cerebral cortex. This is going to include in the caudate nucleus, the putamen, the globus pallidus, the thalamus as well, and the substantia nigra. Now, these names should ring a bell because the caudate nucleus, the putamen, the globus pallidus, and the substantia nigra are part of a system in the brain called the basal ganglia, or the basal nuclei. Now, these basal ganglia or basal nuclei are essential in a couple of functions. One of them is movement. And so Huntington's and Parkinson's are actually quite similar in that they both affect the basal ganglia in some way. They are very different diseases because in one sense they're almost at opposite ends of the spectrum in terms of movement. Parkinson's would be a hypokinetic disease where you're not moving as much. And then Huntington's in most cases is a hyperkinetic disease where a person has uncontrollable moving. Now, as Huntington's progresses, that will eventually stop because enough neurons in the cerebral cortex have died that now the motor cortex cannot send signals down, and then the person is actually immobilized. But that is in the later stages. Okay, so it affects the basal ganglia, and specifically it affects the indirect pathway. So the indirect pathway is going to inhibit, in a normal person, the indirect pathway is going to inhibit overstimulation of the motor cortex, whereas the direct pathway will be stimulating the motor cortex. So if the inhibitory aspect of our basal ganglia is affected negatively, and there's neural death in the indirect pathway, the motor cortex is going to be overstimulated, and that's what causes uncontrollable jerking-like movements. Now, if you take the opposite of that, if you then alter the direct pathway, now you're going to have an over inhibition of the motor cortex, and you're going to be less mobile, and you're going to have issues with movement that'll lead to tremors and then eventually rigidity. Okay, and this is what can happen in Parkinson's as well as happens in the later stages of Huntington's, where there's total neuron loss in the basal ganglia or significant neuron loss. Now, atrophy of the whole brain will also lead to dilated ventricles. And then there's also going to be significant atrophy of the caudate and the putamen, which again have to do with that indirect pathway. A person with Huntington's is going to lose their spiny neurons. These are GABAergic neurons. So GABA is a neurotransmitter. There's also other neurotransmitters that this system uses that are going to be lost in this pathway. But GABA is definitely an important one there. Now, neuronal intranuclear inclusions are also characteristic of Huntington's. These are accumulations of granular and filamentous material in the nuclei of the neurons. And basically, when you accumulate things on the inside of the neurons are not going to be as efficient in processing information. They're not going to be as efficient at sending signals through their axons. There's going to be blocks. There's going to be roadblocks, not allowing cell-to-cell signaling, not allowing materials to be processed within the cell. So those are what neuronal intranuclear inclusions are. So the Huntington gene was actually initially called interesting transcript 15, but we found that it showed mutational differences between Huntington's disease state and normal people. So eventually IT15 was renamed HD, and its gene product was called Huntington. So in patients, there's a trinucleotide repeat, okay? So if you guys remember anything about Huntington's disease, make sure that you remember that it's a trinucleotide repeat disorder. That means that 3 nucleotides within this gene are repeated over and over and over again. And the more repeats that you get, the more you're at risk for Huntington's disease-like symptoms. And once you've already passed a certain threshold, the more copies you have, the earlier you can actually get Huntington's. Okay? So usually there's less than 35 trinucleotide repeats of the nucleotide or the trinucleotide CAG or CAG. If you have 35 or more CAG repeats in this gene, then you're at risk for Huntington's significantly. Now, if you have 65 and more, or 60, more than 60, that's going to lead to the disease presenting before the years of 20, which is very, very early. Because remember that the initial year, or the approximate year in which the symptoms for Huntington's begin to unfold is usually 35, or somewhere around 35. Okay? So therefore, Huntington's disease is the result of an increased length of a trinucleotide repeat. Now, CAG actually codes for an amino acid called glutamine. So you might hear Huntington's disease be called a polyglutamine disease, meaning that the protein Huntington is going to have more glutamine than it should, and that's going to cause these genes to cause the harm that it does. So not only is Huntington's disease a trinucleotide repeat disorder, but it is also a polyglutamine disease. Now here are some images of a normal brain compared to a Huntington's diseased brain. Notice the atrophy of the gray matter, and notice the disorganization now. Also, probably the biggest thing that you see is the enlargement of the ventricles. So that empty space is now larger in the brain because there's so much death. So if you go to the next slide, here's a brain that is Huntington's diseased state. And again, you can see the enlargement of the ventricles, as well as if you've seen enough brain slices, you know that there's a lot of atrophy and disorganization of the gray matter. Okay. Now let's move on to Alzheimer's. So Alzheimer's disease is the most common form of dementia. So dementia is a big umbrella term, and Alzheimer's happens to be at the top in the middle of that umbrella. Now, Alzheimer's is usually really rare before the age of 60, but it can be very common after. So nearly half of people over 85 have Alzheimer's. Now, the biggest risk factor for Alzheimer's, and this might be a little surprising, is actually age. So the older you get, as you get older, the higher your risk is of developing Alzheimer's. That means that as life expectancy increases, this is going to become a larger problem because more people are going to be diagnosed and develop Alzheimer's. Also consider that by 2050 and by later years from now, our population is going to double. So we have over 7 billion people on this earth. And in only a few more decades, we're going to have over 14 billion people on this earth. And at that point, the life expectancy, if it remains the same or increases, the amount of people that are going to be diagnosed with Alzheimer's is going to skyrocket. And currently, the cost of Alzheimer's is about $100 billion annually. And this is because of medication. It's because of what it does to the economy, because people are no longer able to work or be functional units of society. And also because a person with Alzheimer's eventually is going to need care. So they're not going to be able to take care of themselves anymore. They're not going to be able to take medication. They're not going to be able to monitor themselves or go to the bathroom or remember things. And so they're going to need extensive care, and that costs a lot of money, unfortunately. So the cost of Alzheimer's is going to skyrocket as well unless we come up with some type of cure or preventative measure. Now, in the early stages of Alzheimer's, we're going to see forgetfulness, disorientation, difficulty concentrating, calculating, and speaking. So forgetfulness, as we age, we are normally going to lose a couple of brain cells as we age. That's just the normal part of life. But in a person who has Alzheimer's, the amount of forgetfulness is going to be significant of a person that is the same age, or of many people of the same age. So not only are they forgetful, but they are more forgetful than they would be at that age if they had not had Alzheimer's. So keep that in mind. Disorientation. So a huge part of our memory is spatial memory. So the reason that you can come to school every day and find your classroom without a problem is because you remember what's around you. You remember the spaces that you're surrounded by. Going building to building, up the stairs, down the stairs, in one room and into the other, going to the bathroom. You remember where the bathroom is from here because you remember going there. You remember looking for it at one point. So a person with Alzheimer's who's becoming more forgetful is also going to lose aspects of that spatial memory. And so they're going to be very disoriented. Now, in the later stages, they're going to have a significant disturbance in their behavior. So a person who you might remember to be very nice and gentle may be more aggressive and hostile. Psychosis, which is a disconnect from reality. This is very characteristic of someone who has severe schizophrenia, for example. And then finally, they're going to be incapable of self-care, and they're going to become bedridden, and they're going to need more attention from a medical professional. This also becomes an issue because a person who is diagnosed with Alzheimer's early on may be given medication. However, the main symptom of Alzheimer's is memory loss. And so, can you count on a person with Alzheimer's to remember to take their medication? The answer to that is no. And so it becomes very difficult to treat within that aspect as well. Now, in early stages, diagnosis can actually be made pretty accurately, about 80%, and that's in this day and age. If you wind the clock back 20, 30 years, we didn't really know much about Alzheimer's or how to differentiate between Alzheimer's and other diseases, and misdiagnoses were actually very common. Now, in the later stages, accuracy can be even higher at specific centers, and that's because our diagnostic tools are becoming more sensitive and more accurate. Now, doctors are going to look at a patient history. They're going to take and look at neurological exams, psychological exams. They're going to do some lab tests and potentially some brain imaging while the patient is alive to make sure that that person has Alzheimer's and not something like Huntington's or Parkinson's or some other type of dementia. Okay. Now, the final confirmation of whether or not a person had Alzheimer's can only be done upon an autopsy, which means the person would have to already be dead. Okay. Now, when that person dies, or when a person with Alzheimer's dies, and you look at their brain, you're looking for two significant things. One are beta amyloid plaques. Now, the research is up for debate whether or not beta amyloid plaques cause Alzheimer's, or if Alzheimer's leads to the buildup of beta amyloid plaques. It may be a mixture of both, and it may be a case-to-case scenario. We don't really know. But most of the research that I know of is showing that Alzheimer's causes the buildup of beta amyloid plaques. But they might go hand in hand, so we can't ignore both options. The second thing that you're looking for are neurofibrillary tangles. So the beta amyloid plaques are going to be outside of the cell body of the neuron, whereas the neurofibrillary tangles are going to be within the neuron itself. These neurofibrillary tangles are made up of a protein called tau, T-A-U. And they're basically going to disrupt the inner part of the neuron and not allow for cell signaling and the cascades of protein synthesis and things like that are happening within the cell. So understand that, Alzheimer's being a characteristic of Alzheimer's being memory loss, there's a lot of things or a lot of neurological conditions that could also lead a physician to think that the disease is Alzheimer's when in reality it's something else. That can include Lewy body disease or Lewy body dementia, frontotemporal dementia, Parkinson's. A person could have a stroke or have a tumor in the same areas that are associated with memory and have similar deficits. Also sleep disturbances. And there also may be some side effects from medications they're already taking or even potentially an infection. This is why patient history is crucial to diagnoses. So let's go to the next slide and look at the healthy brain versus advanced Alzheimer's. You might notice that the brain looks very similar to a Huntington's disease type of brain. And it's because they're very similar. They're going to have a lot of neurodegeneration within the cerebrum. However, the areas that they take from first are slightly different. And the stages are slightly different. And the deficits are slightly different. So Alzheimer's isn't really going to have significant motor dysfunction. A person will likely still be able to speak and move their arms and legs, but they won't be able to think as well. They will be disoriented, forgetful. They will have trouble calculating, handling finances, etc. Whereas a person with Huntington's disease, some of the first things that you'll see is motor dysfunction. So again, chorea. Now, the known mechanisms, we know that in Alzheimer's, there's a lot of neurotransmitter levels that are reduced. This includes something like acetylcholine. Now, acetylcholine is crucial for the formation of memories and understanding things. So one of the treatment options, medications, is going to affect the level of acetylcholine we have in our brain. So acetylcholine in our brain is normally sent out and uptaken by the postsynaptic neuron, or the next neuron in the link. But excess acetylcholine or extra acetylcholine is then going to be degraded or taken apart, taken down by an enzyme called acetylcholine esterase. Anytime you hear a name end in ACE, it's usually an enzyme. So acetylcholine esterase, or ACHE, as you might see in some textbooks, is going to break down acetylcholine. So one of the medications that we give to patients with Alzheimer's are acetylcholine esterase inhibitors. Because if you can create a drug that is going to inhibit the enzyme that breaks down acetylcholine, you are going to now have more of a sustained level of acetylcholine because you're not breaking it down. So acetylcholine esterase inhibitors have been used for some time now. So you're looking for beta amyloid, which is caused by something called the amyloid precursor protein. So APP for short, amyloid precursor protein. The chromosome for amyloid precursor protein is on chromosome 21. So that gene is on chromosome 21. Now, my question for you guys right now is, do you guys know any other disease that has to do with chromosome 21? If you said Down syndrome, you'd be correct. So another name for Down syndrome is trisomy 21, which means 3 copies of the 21st chromosome. That means that a person with Down syndrome is going to have 3 copies of APP, or the APP gene. This means that they're going to have an overproduction of this protein because there's more gene expression now of that protein. That increases the probability of this amyloid protein to be cut by something called beta secretase and cause beta amyloid to be formed. Normally, alpha secretase and gamma secretase cleave this protein. But when beta secretase cleaves this amyloid protein, you create beta amyloid. And the accumulation of beta amyloid plaques, or the accumulation of beta amyloid proteins, create what is called a beta amyloid plaque. And at certain levels, beta amyloid plaques are toxic to the neurons. So that's one thing. But the second thing is that these beta amyloid plaques are also going to take up space, and they're going to push neurons away, and they're going to disrupt synapses. So think about that extracellularly. So people with Down syndrome are significantly at risk for Alzheimer's, and they tend to develop it earlier because of the probability of forming beta amyloid plaques. Now, another thing to be aware of is apolipoprotein E, or APOE for short. So this is a gene that there are 3 alleles that we know of. So there's 3 different forms. If a person has APOE4 on both chromosomes, then they're at huge risk for developing Alzheimer's later in life. Whereas if you have any mixture of the variants or any of the other variants like APOE2 or 3, those are going to be more protective against Alzheimer's. But again, as you age, your risk is going to be higher because age is the biggest risk factor. Other risk factors includes diet. So what a person eats and how much a person eats will also be very important. The quality of the food, the amount of nutrition in their food, that matters. And 2 is going to be whether that person was very disciplined in their exercise. Did they move or were they sedentary? As a personal trainer, I can guarantee you that if your diet is not where it needs to be and you're not moving at all, you're going to develop things like heart disease, obesity, potentially Alzheimer's in the future. The one absolutely true thing that we know is neuroprotective is exercise. So regular exercise will keep your brain healthy. So that's something to think about. Make sure you're eating your veggies and make sure that you're exercising regularly. Now, the treatment I already gave you guys was potentially acetylcholine esterase inhibitors. On the next slide, we have a couple of FDA-approved drugs and how they work, but I'll let you guys look at those on your own time. Most of the research, again, is pointing to healthier lifestyles are going to help prevent Alzheimer's from developing. So next on our list is ALS, amyotrophic lateral sclerosis. It's also called Lou Gehrig's disease. So Lou Gehrig was a baseball player, and he ended up being diagnosed with this disease. And so he became famous for having this disease, and so another name is Lou Gehrig's disease. Now, this is the most common disorder affecting motor neurons, and it's going to affect 5,000 to 6,000 Americans per year. About 30,000 Americans have ALS at any given time, which means that right now about 30,000 people have this in the United States. The average survival time, and this is the scary part of this disease, is about 2 to 5 years after the person starts presenting symptoms. Now, there are some cases where people can live much, much longer. In about 10% of ALS patients, they can potentially survive to over 10 years after the symptoms are presented. The best example of this is Stephen Hawking. So Stephen Hawking was a physicist that was absolutely incredible as a scientist. He contributed so much to the scientific community, both in literature, in publications for research, as well as influence in pop culture. So Stephen Hawking was amazing. One really important thing to note about this disease is that it only affects motor neurons. So someone like Stephen Hawking, who is arguably one of the most intelligent people to have lived on Earth while he was alive, only lost the ability to move. He didn't lose any cognition or the ability to think and solve problems. And that's a really important distinction that you guys should be aware about when you compare something like ALS to Alzheimer's. So, ALS in terms of diagnoses. So there's no specific test for ALS, like there's no CT, MRI, blood studies. You can use all of these tools to help you rule out other diseases. But your best bet is probably going to be something like a muscle biopsy or a nerve conduction velocity test. If you have issues with motor neurons and they're dying, the signal is going to be a lot less because the person is going to be left immobile. So if you can ask a person to move and test the nerve conduction to the nerves that are going to the muscles, you can see if there's anything wrong there. But there are other motor neuron diseases. This was just the most common. So there are a lot of rule outs that you have to make. Now, the first signs that you usually see are in the extremities. And this is true for a lot of things. So remember diabetic neuropathy? Those are something that you first start to feel in the extremities. The extremities are the weak points, the fingertips and the tips of your toes. So you're going to get something like progressive paralysis in a patient that has ALS. They're going to have weakness in their legs. They're going to eventually develop difficulty walking. This also includes the mimetic muscles of the face. So they're going to have slurred speech. Eventually, all of the voluntary muscles are going to be affected. And this can potentially include the diaphragm. So what is the diaphragm? So the diaphragm is a muscle that lines underneath the lungs, and it's going to pull on the base of the lungs, and it's going to create the negative pressure that you need to inhale. So the only reason that you can breathe is thanks to your diaphragm. And so if that muscle is then impaired, or if the phrenic nerve that innervates the diaphragm is impaired, the person will have issues breathing and potentially they will die from that. Now, another way the death can come is again through pneumonia. Okay. So what are the mechanisms of ALS and what do we know about it? So the neurons that control voluntary movement are lost, but the rest of the mechanisms are really poorly understood. We don't really know what really causes ALS. There are a lot of theories there. But there's no significant genetic component. So 90% of ALS cases are actually sporadic, which means that there's no history of it. There's no genetic component that we've found yet. It's just random and it just happens, which is very scary. Other 10% is inherited. So this is familial ALS. But we have found some genes that have some function in the mechanism of ALS. One of those would be superoxide dismutase. It's just very difficult to assume that this gene would only cause ALS and not any other disease. So it's just very difficult to rule out. And again, it's just very poorly understood. Now, superoxide dismutase, or SOD, is an enzyme that defends against oxidative free radical species, or oxidative stress. So a mutation in the gene that helps you regulate oxidative stress could lead to damage that will lead to ALS. But in my opinion, that's a little bit of a stretch, because that could also lead to other things as well, damage elsewhere in the brain. So it's not as pinpoint accurate as we wish it would be. So that's superoxide dismutase. Now, the treatment for ALS, there's really nothing that we can do to slow down the progression of the disease. The best advice that we could give is just, be as cognitively active as you can be, which means to continue to solve problems, continue to work, continue to do your everyday life. Don't just sit around and do nothing. We can try to lessen the symptoms of it. So we can try to lessen muscle spasms and lessen pain with drugs. But the only drug that can reduce some of that progression is drugs that reduce glutamate levels. But that's about it, and it's very, very small in its effects. So one of the treatments of ALS would be a little bit of physical therapy. So this goes with the idea, if you don't use it, you lose it. So if you use your muscles less, then your atrophy rate or your rate of progression may be larger. And so something like physical therapy might be useful in someone with ALS. Now let's talk about Parkinson's. So Parkinson's is also progressive, and it is a neurodegenerative disorder, but it is characterized by the death of specific neurons, which are the dopaminergic neurons in the substantia nigra pars compacta. So the substantia nigra has two parts. It has the pars compacta and the pars reticulata. The pars compacta is going to be where the dopaminergic neurons are, and the pars reticulata is where the GABAergic neurons are. Now, we care about, in this case, more so the pars compacta and the dopaminergic neurons of the substantia nigra. So, again, age is the really only known risk factor. And some of the characteristics of Parkinson's or PD is going to be slowness of movement, rigidity, some type of gait disturbance or walking and balance impairment, as well as resting tremors. And we'll get back to that. So the dopamine producing cells in the midbrain are going to be the ones that die. So that's within the substantia nigra. If you look at the picture, you'll notice that kind of black purplish mark in the midbrain. So this is a transverse cut of the midbrain. Now, the substantia nigra literally means black substance. And so this is a hyperpigmented region within the brain. So that means that there's a lot of color. There's a lot of pigment there to give it its color. When about 40% of the cells are lost, a person will begin to show symptoms. And that means that up to that 40% of cells being lost, the brain has a way of compensating for the loss of some dopamine. But once that reaches a threshold, in this case 40%, about 40%, the rest of the body just can't compensate and symptoms are going to appear. Now, the cause of Parkinson's is still unknown. It could be environmental and genetic, but we're still considering a bunch of options. So we just really don't know the root cause. Now, within Parkinson's disease, there's the idea of an imbalance of dopamine and acetylcholine. So dopamine is going to stimulate the direct pathway, which is going to stimulate the motor cortex, which is then going to send signals down to your muscles. So if you have lack of dopamine, you're going to get an excessive amount of inhibition, an overactive amount of inhibition from the indirect pathway, and you're going to end up being slower or not moving as much, hence rigidity. So loss of up to 80 to 90% of dopaminergic neurons, and you're going to have a significant amount of symptoms relating to Parkinson's. Another characteristic aspect of the pathology of Parkinson's are Lewy bodies. So Lewy bodies in the substantia nigra are something that you want to look for post-mortem to make sure that that person was diagnosed correctly. However, Lewy bodies can also cause dementia, and that's called Lewy body dementia. So a person with late Parkinson's may also exhibit some signs of dementia that may be Alzheimer's-like. In the next slide, you'll see a couple of real cuts of a real brain. And then you can see how lessened that hyperpigmented region is. So those neurons have died, especially on that top slab up there. Now, on the left of this image, you see what we call this dopaminergic pathway. This is the nigrostriatal pathway that's ascending towards the rest of the basal nuclei and up to the motor cortex. From there, the motor cortex will send a signal down through a descending pathway through the spinal cord to the muscles. Now, if anywhere in that pathway is disrupted, then you're going to exhibit some type of Parkinsonian-like symptoms. Now, in the next slide, we have the symptoms of Parkinson's. And many of you guys already know it. We've already mentioned a few of them. So the way that you memorize this is by memorizing TRAP. T-R-A-P. T is for the tremors, which are resting tremors. This can be in the face, the hands usually, as well as the jaw. R for rigidity or stiffness in the limbs. So this will be walking imbalances. So if your knees are very stiff, you're going to be limping a little bit. Akinesia for A, which is lack of movement. So kinesia or kinesiology, for example, is all about movement. Kinesiology is the study of movement. A, when you start with A, it means lack of. So akinesia is lack of movement. And then P is postural instability. So the inability to maintain your balance and your posture is characteristic of Parkinson's. Now, if we continue down, Parkinson's is treated in a couple of ways. The most common way is by giving the patient two drugs. Levodopa, which is L-DOPA for short. This is an exogenous source of dopamine. It's basically, you can consider it artificial dopamine. The only problem is that if you take levodopa, usually it'll get degraded before it actually passes the blood-brain barrier. So a secondary drug that is used in conjunction with levodopa is carbidopa. And carbidopa is going to stabilize levodopa until it passes the blood-brain barrier. So normally those two are given together. Now, the second type of treatment that we have here is pallidotomy. So the globus pallidus and the pallidum and the subthalamic nucleus is shown to be overactive in people with Parkinson's. This again goes to the dopamine acetylcholine imbalance that we have. So because there's a lack of dopamine, other regions are going to be hyperactive because there's a yin and yang relationship here that we see. And so one way that you can lessen the symptoms of Parkinson's is by deactivating those overactive structures to retain that balance or regain the balance that you already had. Now, this can be contraindicated depending on the specific patient's case. Normally, you don't want to go into the brain so deep just to reduce symptoms. Like there has to be a really good reason for you to pry open the skull of someone and then burn or destroy a piece of their brain. So usually that is only if the symptoms are very severe. Now, next to that, we have DBS, or deep brain stimulation. This is a growing type of treatment for many neurological diseases, including something like migraine headaches. So DBS is actually quite useful, and in many cases we know why it works, and in other cases we are not so sure as to why it works. But this is done by sticking a probe or an electrode very deep into the brain, in this case in the part of the basal ganglia, and then stimulating it with electrical signal. And that can also lead to lessening of symptoms. And then the final case that we're going to consider here is stem cells, or are stem cells. Stem cells have the potential to regenerate these dopaminergic neurons. However, implanting stem cells very deep into a person's brain can be very difficult. because not only do you have to implant these cells, but you also have to direct them to the correct location. It's not like you can just cut or stick a needle in someone's midbrain, because you may be causing significantly more damage than a person with Parkinson's would already have. So there's a lot of things to consider here. Now, one little fun fact for you guys is that caffeine, like what you find in coffee or in energy drinks, and smoking, cigarettes, has shown some protective effects of Parkinson's. Now, that doesn't mean that I'm telling you to go home and smoke and drink coffee all day. Please don't do that. What that means is that there are certain aspects of caffeine as well as nicotine in cigarettes that have some type of preventative or protective effect for Parkinson's. And that might be how those two drugs stimulate the brain. Now, what's also important to note is that if a person has already developed Parkinson's, supplementing with caffeine or more caffeine than usual and beginning to smoke are not going to do anything, because those cells have already died. Now, the last diseases that we have to consider are prion diseases. So prion diseases are misfolded proteins causing a domino effect or a negative cascade effect of misfolding other proteins. And that will lead to neural degeneration. Now, mad cow disease is probably the best example I can give you. So mad cow disease is a prion disease that affects cows. However, humans can get prion diseases as well. So an example of this would be Kuru, Kuru. Now, you might be asking, how do you actually get Kuru? And don't worry, you definitely don't have to worry about getting Kuru anytime soon. Kuru was first found and described by a tribe that had a tradition of eating their past relatives' brains. So, an ancestor to them, or a great-grandfather, for example, once they died, the young ones and the family would consume their brain. And this tribe thought that that would be an honorable thing to do, a way to represent the gaining of new knowledge and continuing on knowledge. Now, the problem with that is that by consuming this brain, they consumed these misfolded proteins, and then eventually they developed certain symptoms. And the onset for this disease, it actually takes 20 to 40 years to see symptoms. So you could live an entire life and then completely go crazy once the symptoms start to present. This includes psychosis, behavioral changes, lack of cognition. So important things about Kuru is that the development of disease is actually incredibly slow. But once it happens, it's like getting hit by a truck. It happens all at once, and then it gets worse and worse and worse very quickly. But the only way you get Kuru is by eating a diseased brain. So please do not eat anybody's brains anytime soon, or any other brain anytime soon. So I'm going to leave you guys with a couple of questions. Question number 1 is, what is a significant difference between Huntington's disease and Parkinson's disease? And why do you see this significant switch in symptoms? Question number 2 is, what is characteristic of Alzheimer's disease? When you cut open a brain of someone who died of Alzheimer's, what are you looking for and why? Question number 3 is, what is ALS, and does ALS lead to deficits in cognition? Yes or no, and why? The final question I'll leave you with is, how much does Alzheimer's cost annually? And why is this becoming an increased problem? Why are we paying so much attention to Alzheimer's today? Why is so much of neuro-related research towards Alzheimer's? Explain. With that being said, I want you guys to discuss that with each other, discuss it with your teacher, think about it at home, come up with a sheet of answers, and then make sure you know those for the Central Florida Brain Beat competition. So this has been the neurodegenerative disease lecture. I hope you guys enjoyed, and we'll see you guys again in the next lecture.